The overall purpose of meiosis is to reduce the normal diploid cells, which have 2 copies of each chromosome per cell, to haploid cells called gametes, which have 1 copy of each chromosome per cell. In humans, these special haploid cells resulting from this process are called eggs in females or sperm in males. Variety can be accomplished in gametes during fertilization. During fertilization, each parent randomly contributes a unique set of genes to the zygote. While fertilization is not part of meiosis, it depends on meiosis creating haploid gametes. The fertilized cell restores the diploid numbers. Without meiosis, the number of chromosomes per cell would double in each generation of offspring, leading to unstable conditions that could threaten the viability of a species.
In interphase, the cell replicates its chromosomes, and each chromosome has 2 sister chromatids held together by a centromere. In prophase 1, chromosomes coil up and a spindle forms. The homologous chromosomes come together matched gene by gene, forming a tetrad. Crossing over may occur when chromatids exchange genetic material. This occurs 2 or 3 times per pair of homologous chromosomes. Crossing over will result in a new combination of alleles on a chromosome. The cell spends most of its time in prophase 1. During metaphase 1, the centromere of each chromosome becomes attached to a spindle fiber. The spindle fibers pull the tetrads to the equator of the spindle. The homologous chromosomes are lined up side by side as tetrads. During anaphase 1, homologous chromosomes separate and move to opposite ends of the cell. The centromeres don't split. This ensures that each new cell will receive only 1 chromosome from each homologous pair.
Non-disjunction is the failure of homologous chromosomes or sister chromatids to separate properly during cell division. Non-disjunction results in daughter cells with abnormal chromosome numbers. This can result in birth defects such as Down syndrome.
During telophase 1, the spindle breaks down and the chromosomes uncoil. A nuclear envelope starts to form around these. During cytokinesis 1, the cytoplasm divides to yield 2 new cells. Each cell has half the genetic information of the original cell because it only has 1 homologous chromosome from each pair. During prophase 2, a spindle forms in each of the 2 new cells and the fibers attach to the chromosomes. During metaphase 2, the chromosomes are pulled to the center of the cell and line up randomly at the equator. During anaphase 2, the centromere of each chromosome splits, and the sister chromatids separate and move to opposite poles. During telophase 2, the nuclei reform and the spindle breaks down. During cytokinesis 2, the cytoplasm divides, and it's identical to mitosis.